dead body and we want you to investigate and find the culprit.
Oh look, Kareena, I found a cigarette.
Let's take it to the lab. What we're going to be doing right now is extract the DNA from the evidence, and then we're going to perform forensic DNA analysis on it. Forensic DNA analysis is when DNA is used for legal proceedings.
This way, we will link the DNA sequence we found in the saliva from the cigarette to the fugitive and hand it to the police officers so they can capture the criminal. Hey, Kareena, I extracted the DNA from the saliva.
That's great. Now all we have to do is put in the PCR. PCR stands for polymerase chain reaction, and it's a 3-step process which creates over 1 million DNA sequence copies in just a couple hours.
So first, we are going to denature the DNA in order for the double strands to separate. This is done by heating up the DNA to 94 to 96 degrees Celsius, which then causes the hydrogen bonds to break between the purines and the pyrimidines.
Second, we will be annealing the strands, which is done by cooling the PCR at 50 to 60 degrees Celsius. This allows the DNA primers to synthesize each single strand along with them attaching to 2 ends of the targeted DNA sequence. This ultimately will allow amplification to occur.
Lastly, the DNA strands go through elongation where the 2 new DNA strands are synthesized at 72 degrees Celsius. The strands that were separated during denaturation then act as templates as the DNA polymerase binds them. A specific DNA polymerase called Taq polymerase is used as it is extracted from a bacteria called Thermus aquaticus, which is found in the hot springs. As this enzyme is from the hot springs, it can withstand high temperatures.
Did you know that Carey Mullis of the Cetus Corporations in the United States discovered PCR in 1983? Because of this innovation, he also won the Nobel Prize in Chemistry in 1993. The idea of PCR came to him randomly as he wanted to observe individual sections of the genetic code. It took him time to convince his colleagues about the idea, but they eventually agreed and helped him create a method. From this, they created a machine that would make millions of copies of DNA by adjusting the temperature itself, and since then, this technique has been used for forensic analysis. But before PCR, another technique known as restriction fragment length polymorphism, or RFLP, was used. RFLP is used to analyze small differences that could exist in DNA sequences, and these comparisons are then used to solve forensic ambiguities. In order for this process to occur, restriction enzymes need to cut the DNA fragments, which later on is compared. After the restrictions have made the cuts in the DNA sequence, other techniques such as gel electrophoresis and hybridization, which will later be discussed, are used to analyze the size of the fragments.
Wow, Kareena, that's so cool! But did you also know modern forensic DNA typing was invented by Professor Alec Jeffreys, which happened by pure accident. He at first started a research program in 1980 to detach the mini-satellites, which are tandem repeats in the DNA from the human genome. During the research, Professor Jeffreys found that a short sequence was shared amongst several other human mini-satellites, which brought up the possibility of mass isolation of highly variable tandem repeat markers, according to Alec J. Jeffreys' article. From then on, he began his work to develop more genetic markers which carried more information than SNPs, also known as single nucleotide polymorphisms, which its marker from the DNA used to be picked up from the RFLP. His main drive to work was for his research to be used in the human medical genetics community, which then led to his discovery in 1984. As he continued his work, an experiment was done in which a hybridization probe that consisted of shared tandem repeat sequences could recognize mini-satellites in the human DNA. However, by pure mishap, that led to him to encounter a method where individuals could be identified through DNA fingerprinting. The first time in which DNA fingerprinting was used was in a case he worked alongside with the FBI, which was a rape and murder case that occurred 3 years apart. Using forensic DNA analysis helped Jeffreys find the real perpetrator of the 2 crimes, who was Colin Pitchfork, which provided an effective demonstration of the method's potential.
Professor Jeffreys' original approach to DNA profiling was by using RFLP, but this process required a larger sample size, which was almost impossible to obtain from a crime scene. As a result, the technique of PCR was later on evolved as it needed a smaller sample size to complete the same task. Since PCR had the ability to test on smaller and older samples, it then progressed on to the future and is more often used than RFLP for forensic analysis.
Before Jeffrey switched to an easier method of using the PCR, his original approach was at first extracting the DNA from the evidence such as hair, skin, or blood cells. The DNA was then cut at specific points along the protein strands and restriction enzymes, which were then placed on a gel electrophoresis. The sorted double-stranded DNA fragments then went through a blotting technique where they were split into single strands and transferred to a nylon sheet. The fragments then undergo autoradiography and exposed to DNA probes, which causes the radioactive DNA to bound to the mini satellites. An X-ray film is then exposed onto the fragment and a dark mark is produced anywhere a radioactive probe is attached. The marked patterns that are then developed are analyzed and matched with other DNA evidence to find the culprit of criminal cases.
After PCR is complete, the DNA goes through gel electrophoresis. This process uses electricity to separate the DNA fragments according to their size. Gel electrophoresis is used to compare the size of the DNA fragments from an unknown sample to the size of the DNA to a known sample to reach conclusions about the identity of the unknown. In other words, samples found at a crime scene are then matched to other known samples which will then be linked to the perpetrator.
After the fragments are separated, hybridization allows the fragments of the DNA to bind together through radioactive probes. This will then allow the DNA to be analyzed by comparing the sizes and distances traveled by the fragments.
Gel electrophoresis plays a vital role in forensic DNA analysis, as without it, it would be difficult to compare DNA fragments. But if it weren't for British biochemist Oliver Smithies in 1950, we wouldn't have had this wonderful invention. Smithies wanted a way to match DNA which was both fast and cheap. Scientists were doubtful about his idea as it was something that was never done before, but it later on became the greatest invention of his time and is still widely used today for forensic analysis.
DNA profiling by law enforcement was first used for identifying rapists, murderers, and other atrocious offenders. The first time DNA test results was used in a criminal trial in the United States was in 1987. Although DNA test results were relevant evidence in criminal cases, historically, courts restricted the DNA results' admissibility due to the reliability of the testing process. It wasn't until 1994 when the Supreme Judicial Court established a valid admission of statistical probability in the match from the DNA tests. The use of DNA profiling became popular around 1990 in the Western Balkan region due to the identification of human remains from mass graves. Other smaller countries such as Bosnia and Herzegovina, Croatia, and others also began the use of forensic DNA analysis of skeletal remains and databasing their results. Since 9/11 in the US, the law enforcement agencies around the world expanded the areas of forensic DNA databases due to the terrorist attack. As the forensic genetics started to expand, organizations began to get concerned on the sociological perspective of DNA profiling.
As the DNA samples are kept in databases, personal information about an individual's characteristics, family, and certain diseases is most likely gathered. This causes an intrusion of privacy when the law enforcement interrogates family members of a suspect. Therefore, requesting for the suspect's DNA can negatively impact their life through disrupting their career or marital life. Many say this is a form of harassment as it violates a person's privacy, as according to a law professor, Sonia M. Sutter, it could subject them to a lifetime of genetic surveillance.
In 2008, the European Court of Human Rights ruled that DNA samples and profiles of a suspected person who was not convicted of any offenses violated the right to privacy. Laws have been made throughout the years where DNA samples were to be destroyed of a person who was not charged or was innocent in the UK under the Police and Criminal Evidence Act. However, that was amended by the Criminal Justice and Public Order, where DNA samples were allowed to be retrieved even when charges were dropped or not proceeded, which could then be used for future investigations.
From 2003 to 2005 in the United States, the U.S. National Commission and the American Society of Law, Medicine, and Ethics Project constructed a consultation to present to the government about the ethical and legal issues that consisted of questions about privacy and civil liberty. It talked about changes in the government research and operational issues of forensic genetics by a human rights-based approach on public protection and security.
Look, I found out who killed Mr. Bear.
Oh, that's great!
Let's take this to the FBI.
How did you find the culprit?
I performed forensic DNA analysis by first completing PCR and then gel electrophoresis.
But Sukhleen, who is it?
It's Mr. Bear's brother.